I'd like your attention for just a moment. Angie asked me to make an announcement, which you can read on the screen overhead, and I'll email it as well, but I didn't want anybody to come out in this kind of weather and be disappointed. So she has medical conditions that's making it impossible to do it tonight, but should be back by next week. And with that, I will turn it over to Carissa. Let me just email it. All right.
Okay. All right. Well, I want to thank you for coming back for my second lecture in this class. I appreciate seeing people who were here on Wednesday and actually want to return for another lecture, especially with the weather so icky. So actually, I was just remembering when I was an undergrad and weather like this, when you would often want to just stay home and take a nap, or rather be home and be warm. I remember my undergraduate advisor who would often come in to the lab and say, "It's a great day for science, isn't it?" And so, I have to admit, I say that to myself once in a while when I don't want to go to work. So it's a great day. Let's get started. Learn some science. So just as a quick review, so we're all on the same page from where we were and where we stopped on Wednesday. We started out looking at different structures of DNA, the DNA topology, and also some rearrangement of the topology. And then we started moving into the process of DNA replication and enzymes and proteins that are involved with replication. So if you remember, we looked at helicase, which unwinds DNA, single-strand binding protein, which binds to single-stranded DNA and keeps it from interacting with other possible things that's found in the cell. And we also looked at sliding clamps, which served as a bit of an anchor for DNA polymerase to grab onto the DNA and facilitate replication. And so with that, I'm going to continue on talking about DNA polymerase. I think we stopped right on this slide on Monday. And we were talking about the structure of DNA polymerases. Now it turns out that all DNA polymerases have very similar structure. If you can look at the X-ray crystal structure of these proteins, they all have what resembles a right hand. Now the amino acid sequence might not be the same. Actually, they can vary quite a bit, but they all look very similar in the overall structure. And this hand has fingers domain, and then a palm, and then a thumb domain. So it looks like perhaps, like I said, a right hand, where you can see that you could easily fit some DNA and nucleotides into this area. And it turns out that the palm is what holds the DNA. I'm going to make sure this is set to visible, so you can see this. Okay, so the palm holds the DNA substrate, so the template strand, as well as the newly synthesized, a short fragment of newly synthesized DNA as well. It also has magnesium, which is important for the catalysis of DNA synthesis. And the fingers and thumb help to grab the nucleotide and hold it in closely to the DNA substrate for nucleotide incorporation. And as I said, that magnesium helps, as the nucleotide is being brought into the DNA, it helps to align it in the proper orientation so that it's perfectly aligned for incorporation onto the primer strand. And the way DNA polymerase works is basically like an opening and closing hand, where it grabs the nucleotide, holds it in place, allows incorporation of the nucleotide, then moves down one base on the DNA template, grabs another nucleotide, incorporates it, and moves on like that for many, many bases at a time, particularly if the sliding clamp is there to hold it and keep it associated with the DNA. Also turns out that there are many different DNA polymerases. There's as many as 5 or maybe even more in E. coli. And there's at least 15 DNA polymerases in eukaryotes. And these are all categorized into 6 different families of DNA polymerases. But given the fact that you think that replication is simply incorporating nucleotides, you might ask, Why are there so many of these DNA polymerases? Well, DNA polymerase is really good at what it does, but usually it is designed to only do that function. Like many enzymes, they can only accomplish one reaction. So you need, there's many different functions that require DNA polymerase, such as replication of the leading versus the lagging strand of DNA. And we'll look more at these two later, what the leading strand and what the lagging strand actually is. Also, you might remember that mitochondria have DNA as well. So you need DNA polymerase in the mitochondria to accomplish replication of that DNA. The process of sister chromatin cohesion requires polymerases to function in that process. Also, DNA is very susceptible to damage within the cell, and there are polymerases that function specifically for replication through damaged DNA. This is the structure of a DNA polymerase that is specifically involved in replication of damaged DNA. We're actually going to talk about this even more in chapter 18. I believe it'll probably be Monday before we talk about it, but it's really a very unique DNA polymerase, very exciting. Not to mention that I do research on this enzyme, so had to stick that in there. Also, there may be other functions for DNA polymerases that aren't even fully understood yet. There are some DNA polymerases that are specialized, and we observe certain activities for it, but we're not really sure what it actually does inside the cell. So there is a lot yet to be learned about even these DNA polymerases, particularly the eukaryotic DNA polymerases. Now, as I said, DNA polymerases that continually replicate DNA, that are involved with replication of the whole genome, are really good at replicating DNA. This is because mistakes that are made in DNA can be disastrous. And again, I'll refer you to chapter 18 when we're talking about DNA damage and replication and how this can lead to cancer, actually. So as a result, the replicative DNA polymerases, those that catalyze replication of most of the genome, have very high fidelity. Now, fidelity refers to the fact that DNA polymerase makes mistakes very infrequently. So it's very rare that you might find a misincorporated nucleotide if these polymerases are catalyzing DNA synthesis. And there are two methods by which the polymerases actually accomplish or act to prevent mistakes being made or mispairs, incorporation of incorrect nucleotides. First of all, there's the active site for DNA synthesis, and we'll look closely at this to see how it actually helps to prevent mistakes being made. And then secondly, there's a second active site called the exonuclease site, and that serves to proofread the DNA that was just synthesized. So let's start with the active site of the DNA polymerase. Now, this active site is if you remember, it's in the palm of the DNA polymerase. And at the active site, what happens is the incoming nucleotide, this dNTP, is paired with the template and incorporated into the primer strand, which is the growing strand of DNA. And it turns out that this active site is very tight and specifically shaped only for Watson-Crick base pairs. So only the correct adenine to thymine, or vice versa, thymine to adenine, and guanine to cytosine, or cytosine to guanine base pairs fit into the active site. What we have in this picture here is actually 4 different DNA polymerase structures with DNA, the correct Watson-Crick base pairs bound to the DNA. So you can see in white, this light, I guess it's more like a creamish color. These are residues that are part of the DNA polymerase. And in yellow, we have all 4 possible combinations of the correct base pairs. So we have adenine to thymine and thymine to adenine, and the same with guanine and cytosine base pairs. Now, with all of these 4 structures overlaid, if you look at these 4 possible correct base pair arrangements, look at the overall geometry of these base pairs. It really has a very similar structure and a very similar shape. And it turns out that the active site of the DNA polymerase is only able to fit this shape into the active site. So it's basically like a tight door that doesn't allow anything abnormal into the DNA polymerase. So if you add in a mismatch, say you have the adenine base pair to guanine, it actually causes a bit of a distortion in what this base pair arrangement looks like, and it kind of sticks out a little bit more, and it doesn't fit well into this tight active site. So as a result, this different geometry doesn't fit very well. So it's not likely that the polymerase is actually going to try to incorporate the wrong nucleotide. And so we can see that the active side of the DNA polymerase is basically the first line of defense against creating mismatches in the DNA substrate. So the second line of defense against mistakes with DNA is the exonuclease site of the DNA polymerase. As I mentioned, this is a different active site. It's a separate site other than where the DNA is actually synthesized. So if the DNA polymerase does somehow happen to incorporate an incorrect nucleotide, it somehow senses that this mismatch doesn't quite feel right, and it sends the whole strand of DNA to this exonuclease site within the polymerase. What the nuclease does is removes this incorrect nucleotide. It's a 3' to 5' exonuclease. You can see that this primer strand here, this end right here is the 3', excuse me. So it removes from the 3' to 5' direction. So it removes the offending misincorporated nucleotide, sends the DNA substrate back to the active site to give the polymerase another crack at incorporating a nucleotide. And thus, it gets a second chance to do it correctly. Having this exonuclease site in the polymerase enhances the fidelity of the polymerase even more. It's as infrequent as one mistake made by a DNA polymerase for every 1 million nucleotides that it incorporates. So that's a pretty good ratio, considering that DNA polymerases usually incorporate very fast as well. So it's fairly impressive that these polymerases are so good at making the correct base pairs. So far we've looked at DNA polymerase and how it works to incorporate nucleotides and create newly synthesized DNA. Now we're going to look at a few potential problems that the polymerase might encounter while accomplishing replication of the genome. Now, one potential problem is that DNA polymerase needs something to add the nucleotide onto. It needs a 3' OH to incorporate the nucleotide and add it on to. It can't initiate DNA synthesis without anything there. So if you have only a template DNA strand, the DNA polymerase cannot bind to this and just start with replication and continue. So in order to overcome this problem, the cell has an enzyme called primase, which initiates the replication by laying down a primer that is composed of RNA. So it lies down, in red here would represent the RNA primer. So primase lays down this RNA primer onto the DNA, forming a DNA-RNA hybrid. The key is that it provides the 3' OH for the DNA polymerase to start adding nucleotides onto. And in that way, DNA replication is begun. And DNA polymerase can basically grab on and continue with replication after this primer is inserted. Now, we don't really want RNA to be present in the DNA at all times, so actually it turns out that the RNA primer is later removed and replaced with the DNA by a polymerase. Another potential problem that DNA polymerase might encounter with DNA replication is the fact that it can only synthesize in the 5' to 3' direction. Now, this really isn't a problem except for the fact that at the replication fork, we have anti-parallel DNA. So you have one strand that might read in the 3' to 5' direction, and another strand that reads in the 5' to 3' direction. So if we have two DNA polymerases, one acting on each template strand of the DNA, they want to accomplish DNA replication from 5' to 3' with the new strand. And it turns out that they want to go opposite directions. Now, if you remember, DNA polymerases are found at replication factories. And these factories are stationary. So these two polymerases actually aren't moving. It's the DNA that is moving. So it would require some major pulling of the DNA polymerase, or of the DNA itself, in order for replication to be continuous with both of these polymerases. You would have to pull the replication fork in two different directions, which is not easily accomplished. So instead, we have what I referred to earlier with leading strand synthesis and lagging strand synthesis. So the DNA polymerases, here we have a picture of DNA in blue. These two green circles represent the DNA polymerases that are working on at the replication fork. And this polymerase on the left is accomplishing continuous DNA replication on what is called the leading strand. The second polymerase on the right is not going to be able to accomplish replication continuously because it's actually on the lagging strand of DNA. And what happens is as this replication fork unzips, the polymerase going in 5' to 3' direction, as I said, on the leading strand, has no problem. It keeps going along continuously. But since this polymerase on the right is on a strand, it's trying to go 5' to 3' direction, but the replication fork is actually moving the opposite direction. It kind of it accomplishes replication for a short distance, but it also causes this strand of DNA to essentially loop out. And as it loops out, primase, can lay a new RNA primer. And essentially what happens is that after the polymerase has moved far enough down, essentially it jumps to the next primer and accomplishes replication from there for a short distance. And again, as this unzips, this strand up here would begin to loop out and a new primer would be laid down. And the polymerase would essentially jump to the next primer and accomplish replication again for a short distance. So what you end up with on this lagging strand is short fragments of RNA primers with DNA that are discontinuous and are not connected with each other. And these short fragments of DNA are called Okazaki fragments. They're named after the person who discovered the fact that there actually is lagging strand synthesis. Now, from that picture that we just looked at, obviously, on the lagging strand, there's going to be multiple Okazaki fragments present, and lots of strands of short bits of DNA that are not connected with each other. And there are going to be RNA primers that need to be removed as well before the DNA can be made continuous on this strand. So there's an enzyme called RNase H, which functions to remove these primers. So if you look here, we have the template strand of DNA in blue. And here's the DNA polymerase in green. Looks like it's gone from the first Okazaki fragment to the second primer and started synthesis of the second Okazaki fragment. RNase H can come in and remove this first primer that was laid down and thereby allow DNA polymerase to fully synthesize up to the point where the other strand of DNA begins. And as that occurs, after that occurs, what's left is simply a nick, a single strand cut in the DNA, and that can be sealed by DNA ligase. Now you might imagine that DNA polymerase is pretty fast, and if primase doesn't act fast enough, the polymerase is going to encounter RNA primer. What happens when DNA polymerase actually encounters the RNA primer is basically it pushes it out of the way and continues with replication of DNA. So you can see as this polymerase with this picture, as the polymerase encounters this first primer, basically it sticks the primer out like a little flap and continues replication up to the point where the next DNA strand begins. There's actually an endonuclease which is designed to specifically work on this RNA that's sticking out like a flap. And this enzyme is called flap endonuclease, and basically it comes in and it can remove this flap. And you see that we're left with a simple gap that can be refilled with ligase. So we've looked at DNA replication by DNA polymerases, how they work, what potential problems there are for DNA polymerases. Now we're going to move to a different kind of problem which might potentially be encountered with eukaryotic chromosomes. This is called the end replication problem. And this results from the fact that eukaryotic chromosomes are linear. If you remember, bacterial chromosomes are usually circular, so they don't actually have this problem. But with these strands of DNA where replication can occur until the end and stop at that point. It's okay if you're following DNA template where you encounter, as it encounters the 5' end of the DNA template strand. Because DNA polymerase can simply replicate up to the very end and then fall off and leave the DNA as is. But if you look at the opposite end of that template strand, that would be the 3' end of the strand, there's a slightly different problem. Because you needed the DNA polymerase needed the RNA primer to be laid down for synthesis to even be begun. This RNA primer is present on the 3' end of the template strand. Once the primer is removed, there's still a problem because DNA polymerase cannot come in and initiate synthesis to fill in the gap that's left by the primer because there's no 3' OH there. So there's a gap that's left. It's basically a strand of DNA that hangs over, unpaired with another strand of DNA. You might imagine that if you're looking at a replication fork, the 3' ends of both of those strands of DNA are going to be left with gaps on them. It turns out that there's nucleases present in the cell that like to cut up DNA, and they specifically can cut up single-stranded DNA. So if there's 3' overhangs of single-stranded DNA left at the end of the chromosome, these nucleases come in and cut up those fragments, resulting in slightly shortened chromosomes. Thus, if there's no method of protection, each round of DNA replication would result in shortening the chromosomes. Now, you might imagine that it wouldn't take very long before you start cutting into some very important DNA, some important genes that are necessary for the function of the cell. So the cell actually has a way to prevent this shortening of chromosomes, and that's with these structures called telomeres. And they help to prevent this end replication problem of DNA shortening. Telomeres are basically extensions of chromosomes. They are extra repeating units of DNA that are added on to the 3' end of the chromosomes. Now these fragments of DNA, you can see each of these little blocks here in this picture represent one of those repeating units of DNA. They don't really encode anything, so it's not really important for transcription. It's important for protecting the chromosome. But essentially, these telomeres are dispensable. They can be removed without any cost to the cell. So with a telomere present at the end of the chromosome, actually if a primer is laid down, DNA polymerase would replicate the telomere as well. So you would have DNA, after a round of DNA replication, you'd be left with a single strand overhang of telomeric DNA, but the chromosomes are fully double-stranded. So as nucleases cut up this extra DNA, it's really not hurting the chromosomes, it's just removing extra DNA that's serving as a buffer, as a way to protect the important genes. And these telomeres can be extremely long. In humans, it can be anywhere from 5 to 15 kilobases long in human cells. Now, the enzyme complex that actually accomplishes adding on telomeres onto the DNA is called telomerase. The telomerase is actually a complex of many proteins. It's called a ribonucleoprotein because, as the name suggests, there's an RNA component and there's a protein component. The protein component is reverse transcriptase, and many of you might recall that reverse transcriptase is actually a DNA polymerase that can use an RNA template. So basically, telomerase serves as its own template, uses the RNA component to serve as the template, and uses the reverse transcriptase component to add on DNA onto the chromosome. So basically, the telomerase can act on the end of the chromosome at the 3' end to add more of these repeating units of DNA. And this is referred to as RNA template directed DNA synthesis. And that's how telomeric DNA is added onto the chromosomes. So when telomerases were initially discovered, there was actually a lot of excitement because many people thought they had found the key to eternal life, or the fountain of youth, the way to stay alive forever. This is because they observed in mortal cells, cells that essentially die, that telomerase is not active. And telomeres thus would decrease with each round of replication. Eventually, as the telomeres decrease and it gets closer and closer to chromosomes, the cells would begin to reach what's called the senescent stage. This is a stage where they basically stop growing and they don't divide anymore. And the cells essentially, you might say, they aren't alive. And what was observed with cells that are immortal, those which can undergo multiple rounds of replication without any cost, they found that telomerase was actually active. The chromosomes were thus maintained, and the cells could continue living. So people saw this observation that immortal cells had active telomerase. They thought, hey, if we could just activate telomerase in each person, we could stay alive forever. Well, there's maybe some idea there. There's maybe a correlation. We don't know if that's necessarily true. There's some evidence with this bird here, which is called Leach's storm petrel, where actually there's a study done that found that some of these birds can live for a very long time. And it turned out that those that live longer have longer telomeres. So I guess that idea might support the fact that telomerase and telomeres help to lengthen life. But again, it's only an observation, maybe a correlation, but you might remember that correlation doesn't prove causation. You might remember also that aging also involves many other processes and other functions, such as apoptosis, and cancer might play a role in aging and death. There are many other things that can actually affect the cell. So it's not as exciting. Well, it's still very exciting research, but maybe not quite with the same application that many people thought they had found. So we're going to shift gears a little bit now and talk a little bit more about how the DNA is actually packaged into the nucleus. Now this picture here is a picture of a eukaryotic nucleus with different types of DNA stained in different colors. In red, I'm sorry, this arrow should be pointing to heterochromatin. But in red, you can see it's mostly at the periphery of the nucleus. This is heterochromatin. And this is DNA that's tightly packed. It's not transcribed, so it's not actively being transcribed into an RNA or protein. And also, it's near the periphery of the nucleus. Which makes sense, because if the DNA is not being used at that moment, it's easier to simply move it to the outside of the nucleus where not a lot of activity is going on. There's actually correlation between activity, or the amount of transcription of the DNA, and where it is located in the nucleus. The yellow is a picture of euchromatin, so this arrow should be pointing to the euchromatin. I don't know why these are messed up, sorry about that. Anyway, euchromatin is DNA that's looser. It's not as tightly compacted, and it's actually commonly being transcribed into RNA. And as a result, it's found near the center of the nucleus where a lot of the activity of transcription is going on. So you can see that how tightly the DNA is packed affects how active the DNA, how actively it's being transcribed. So let's look a little bit at what this packaging of the DNA actually looks like. Now, the fundamental unit of eukaryotic chromosomes is the nucleosome. The nucleosome is just a repeating structural unit composed of DNA and proteins called histones. And they help to compact DNA. In human cell, you have nearly a meter of DNA if stretched out fully. This compacts all this into a 5-micron nucleus. So you can imagine that there's a lot of compaction that's going on. And it turns out that tightly compacted DNA is not transcribed off as much, as I said earlier. And so compaction of the DNA helps to regulate transcription. You might imagine that if it is really tightly wrapped around these proteins, it's not going to be easily accessed to the DNA, accessed by the RNA transcription factory. So what a nucleosome actually looks like is a component of 8 histones, repeating units of these proteins represented in green, with the DNA in red wrapped around those proteins twice. And this nucleosome resembles what's often called a beads on a string model. So it's composed of the nucleosome core. As I said, there's 8 histone proteins. Here's another drawing of what a nucleosome might look like. In yellow would be the histone proteins. There's 2 copies of each kind of histone. So there's histone H2A, there's 2 copies of H2B, H3, and H4. And these 8 proteins serve as the core for the DNA to be wrapped around. Here's another picture of the nucleosome core, where you can see the DNA wrapped around these proteins. And if you look on the side in a different model of the DNA, or of the structure, you can see the DNA is tightly wrapped directly around these proteins. There are tails on these histones that are important for interactions with DNA. So here's a close-up picture of the nucleosome core. Again, you can see the DNA, and there's 1 copy of each histone. H2A is in yellow, H2B is red, H3 is blue, and H4 is green. If you look at the N-terminus of these proteins, there's these tails. And if you might note, they're all interacting with DNA. And these interactions are very important because there's modifications that can be added onto these histone tails, which affect how tightly the DNA is compacted around the nucleosome. And we'll look a little bit more at histone modifications in a little bit. So the histone core is what we just looked at. That's those 8 histone proteins with the DNA wrapped around it. A complete nucleosome is composed of that core as well as another histone protein called the histone H1. So this picture shows another drawing of what histones, the complete nucleosome would look like, where in red are the 8 histone core proteins. The DNA is tightly wrapped around those proteins. And I usually think of histone H1 as being the paper clip that holds it all together. Basically seals that whole interaction and stabilizes the DNA coils where they are with the histone proteins. So you can see there's multiple repeating units with complete nucleosomes with this histone H1 added on to hold the nucleosome together, essentially. You might imagine that who wraps around these 8 proteins is quite a distance. Actually, it turns out that compared to the length of the DNA fully linear, this structure helps to compact the DNA size by 30 to 40 times. So this is 30 to 40 times smaller than uncompacted DNA. And it turns out that not only are there complete nucleosomes, but there's also higher order of structure of compaction for the DNA. And this has to occur prior to cell division because you don't want big, long strands of DNA sticking out, available for possible problems, or just getting tangled up and in the way when the cell is dividing. So prior to division, DNA is compacted even further. And the nucleosomes are compressed into what's called a solenoid. And this is a picture, a drawing of what it might look like. They're basically extremely short rods of lots of DNA. They can be as short as 10 microns long. As I said, that these shorter strands of compacted DNA help to prevent tangling of DNA with other DNA molecules or other proteins or whatever else might be around. Also, it's easy to move something that's shorter than something that's really long. When it's compacted like this, though, the DNA is not available for replication or transcription. So after cell division occurs, this solenoid has to be unpacked, basically, before any of these cell processes can occur. These solenoids can actually be seen by electron microscopy. And again, you can see the beads on the string model. Here you can see the compaction of the DNA, what looks like little beads on this string of DNA. And how tightly these things pack depends on the environment a lot. You can see that salt, these are in different environments where you see it's tightly compacted, not quite so tight. So salt, histone H1 present in the protein can also affect packing of these solenoids. Modifications on those histone tails, as I said earlier, also greatly affect how tightly these things are compacted. Other proteins present, as well as supercoiling of the DNA, all play a role in packaging of the DNA into these beads on a string model. We're going to move up another level of compaction to a higher order structure, and that's solenoids being packed onto a scaffold or a matrix for mitosis or interphase. So if you look closely, here's a 30 nanometer solenoid that loops around. All of these solenoids are attached to a scaffold. You can see there's just lots and lots of protein and DNA on these scaffolds. It's a good way to definitely compact the DNA. Okay, I'm going to move on to histone modification. As I mentioned earlier, those histone tails which are interacting with the DNA are important because of modifications that can be added onto them. Lysines can have acetyl groups added onto them. They can have methyl groups added on. Serines can have phosphates added on. Phosphorylation can occur on those serines. And those tails touching the DNA affect what happens at the DNA that's right next to it. Basically, these modifications can be recognized by certain proteins or not recognized by proteins to affect the protein-DNA interactions. So this kind of shows here a picture of what might happen, where you have these histone tails that are acetylated and methylated or phosphorylated. And these different additions on the histone tails serve as signals for proteins that can come along, and perhaps one is designed to specifically recognize these different signs that are present on the DNA, and they can come and bind to the DNA because of those modifications. Or maybe the protein doesn't recognize these signs unless they don't interact with the DNA. So the histone modifications are very important for regulation of transcription of DNA as well as other necessary processes with DNA. This modification, as I said, they alter chromatin structure, and the gene expression can be altered specifically by histone acetyltransferases. These are proteins that add acetyl groups onto the lysines. As acetyl groups are added on, it basically opens the chromatin, makes it more loose, more available for proteins to interact. So transcription can actually occur. So here we have tightly compacted DNA with unmodified histone tails in green. When the histone acetyltransferase, HAT, adds acetyl groups onto the lysines, basically the acetyl groups kind of help to separate it all. You might imagine that the DNA is not quite so tightly wrapped around the histones as a result, and thus RNA polymerase can come in and transcribe the DNA. There's also histone deacetylases, HDACs, which serve to remove acetyl groups. So if you have a situation where the acetyl groups are already present, they can be removed with the HDAC to tightly compact the DNA and the chromatin, and thus make this DNA transcriptionally silent because it's so tightly compacted and it's not as easily accessed by the RNA polymerase. Here's an example of what unacetylated DNA might look like in histone compaction, where you see that it's very tightly compacted together and thus silent. Whereas if it's acetylated, these nucleosome cores are moved farther apart and transcription can actually occur. Okay. Now, it turns out there are also higher orders of packing of the DNA, as I said. So if we start from the DNA double helix being wrapped around the nucleosome core, then compaction into a 30 nanometer fiber of packed nucleosomes, which are added on to the scaffold or matrix to compact even tighter, and eventually to form the chromosome, which holds everything together. So we get a lot of DNA compacted into a very tight molecule. Okay, are there any other questions before I move on to DNA modifications? So like proteins can be modified, DNA actually can be modified as well. And it turns out that cytosine can actually have a methyl group added onto it. So here we have a picture of a 5-methylcytosine, where the cytosine has this additional methyl group on carbon 5. And this methyl sticks into the major group of the DNA. And as a result, this methyl group alters interactions between proteins, or could potentially alter interactions between proteins that bind to the major group of the DNA. There are processes which are specifically involved in adding methyl groups onto the DNA. It turns out that if there's a sequence of a cytosine next to a guanine, those are specifically targeted for methylation. Near the start of genes, these CPGs, T simply refers to the phosphate that's in between the two nucleotides. These CPGs near the start of genes are actually unmethylated. And it turns out that this methylation may be a marker for silent DNA. Because when the methylation is there, DNA is not as oftenly transcribed. So modification of DNA also alters how often the DNA is transcribed, just like modification of the histone tails, or in a similar manner. Now, as DNA replication occurs, the modifications, say methylation of the DNA, has to be copied from parental DNA, and it turns out it's copied with the same patterns into the daughter DNA. So if you have parental DNA with methyl groups, here's a CPG. So here's a cytosine and a guanine. And both cytosines in this sequence are methylated. As DNA replication occurs, one parental strand goes to each new strand of DNA. So one strand is methylated properly, while the newly synthesized strand is not. So there are enzymes that are involved with specifically maintaining this parental pattern of methylation at these CPGs, and they help to pass on traits of silenced genes, and thus add the methylation in the appropriate spots in the new DNA. These are called maintenance methylases, and they help to maintain the correct methylation of DNA. This is involved with genomic imprinting, which you might be familiar with. Because of time, we won't be going into that today. There is a problem with methylation of cytosine, and that's the fact that spontaneous deamination can result in cytosine being turned into thymine. So if you lose this amine group here, and it turns into an oxygen, deamination occurs where an oxygen replaces the amine here. This is a thymine base instead of a cytosine. And thus it does result in the transition from a C to G base pair to a T to A. So this is a point mutation that results from DNA modification with methylcytosine. Okay, so we're going to switch a little bit here to talking about chromosomes and bar bodies. Chromosomes are not usually visible during interphase because they're not transcriptionally active at this point. I'm sorry. Give me a second here to figure out what I was going to say. Sorry about this. Okay, so bar bodies are the result of females have 2 X chromosomes, right? You only need 1 X chromosome, really. The other one needs to be inactivated. And it's random decision based on which one is actually inactivated. And this is what a bar body actually is. It was named after the person that discovered them, but often people refer to bar bodies. Remember bar bodies as the fact that they're barred from transcription. So these bar bodies may be the result of hypoacetylation, methylation of CpGs, and also this exist RNA, which serves to coat DNA to prevent transcription of the bar bodies. Now, you might remember that there are some potential genetic disorders that actually could result from bar bodies that could result in difficulty in defining what is male and what is female. For example, Klinefelters, where there's 2 X's and a Y chromosome in a person. There's one bar body present in this Klinefelter syndrome. Turner syndrome has only an X chromosome. No bar body is present, yet it still resembles a female because you only really need one X chromosome. Also, it turns out you can have an XX male, which does not have a bar body at all. An XX male? Right. So an XX male is the result of translocation from a Y chromosome, a gene that's necessary for definition of a male. So it's one specific gene, it's called the SRY gene, that's translocated onto an X chromosome from a Y chromosome. Right.
Put that SRY on the X chromosome.
There's also the possibility of having an XY female, where the same sort of thing occurs, where there's an alteration with one important gene for a definition of a female. And the final thing we're going to look at is called the C-value paradox, where I'm sure many people have come across this before. This is the fact that the total amount of DNA present in a cell doesn't match how complex that organism is. So if you look here, here we have the size of genomes in base pairs, and you can see that humans are up here where the red is shown. You see that flowering plants, some can have many, many more numbers of base pairs than humans, yet they're not nearly as complex as humans. So the C-value paradox refers to the fact that the amount of chromatin present in the DNA doesn't reflect how complex the organism is. We don't know really if it reflects a function or if it's just an accident of what happened through the course of evolution. But see that humans and other similarly related organisms can vary a lot in how much DNA they actually have present in their genes. So I think that's the end of it. So thanks for coming, and we'll see you on Friday. The bar body. What's that going to say? What was my transition?